So thank you very much for your very kind words, Christian, and for the opportunity to be here and tell you a little bit about our research. As Christian said, my group has been interested in the structural biology of tropical diseases, in the sense that we prefer very much to focus our work on proteins which come out of infectious diseases from low-income countries, and then we hope that they are so important that when we inhibit those protein molecules, there is a possibility that further development by chemists and parasitologists, bacteriologists, and further tests will lead to compounds which can finally reach patients. So I'll show you a little bit about drug action and drug design in general, only some very global slides, and then we'll go to some specifics, and we'll see where we are at the moment with one of our projects. So is it okay if I stand in front here? Then I can see better what I'm projecting. So first, how do drugs actually work? And here's sort of a summary of how drugs act. And this is not only how they act. It's with my very good friend Paul Michels, we are giving workshops in low-income countries about drug design for neglected tropical diseases. And this slide comes from my lecture on inspiring modes of drug action. So it's supposed to be inspiring, and also has some very nice attempts which have not yet been successful. But this is a lecture, it's actually two lectures in one, too long in most cases, but I'll spare you all the specifics. So we just go over the general picture. By far most drugs work on proteins, several work on DNA, protein-DNA complexes, protein RNA assemblies. The most famous, of course, is the ribosome. Here's an example where we have no drugs yet to the best of my knowledge. Ribo switches, membranes, there are examples. Very unusual are drugs which works on substrates. But one of them you probably know very well, which is vancomycin, which does work only on a piece, on the ala, as you probably know. And there are small molecules which react with very unusual other small molecules. You must have heard of chloroquine in the case of malaria, and that mode of action is extremely unique. So as you know, malaria eats, the diet of malaria is very simple. It's hemoglobin, but it only eats the globin. So the heme is left over. Often leftovers are okay, but in this case, the leftover heme is very dangerous, because the iron generates oxygen radicals, and those oxygen radicals kill everything. So the parasite has found a way to avoid the toxic effect of the leftover heme by stacking the heme into crystals, into linear arrays, where a carboxylate of one heme coordinates the iron, and in that way we have no dangerous heme anymore. That's called hemozoin. And chloroquine interferes with this stacking of heme groups, and therefore there is free heme still available and generates oxygen radicals and kills the malaria parasite. It's a very unique mode of action, of course not by design, but there are many cases where it's by design or by going after a particular target. So if you look at proteins, many proteins block active sites, but many drugs, many drugs block active sites, but a few activate a protein, so it's too active. So as you know, a cell is a control freak without a control center. So it wants to control everything, but it has no central system to do that. It is all by local feedback loops. And protein activators make a protein too active, and therefore the cell is in trouble. We don't have time to go over all of this. My favorite drug is actually the topoisomerase inhibitors. So ciprofloxacin and also camptothecin in the case of cancer, they have a very unique mode of action. You see, when you block an active site, you almost always have to make sure that the whole flux through that pathway is very small. So you have to have very tight binders. With topo inhibitors, that is not at all needed. It's an entirely different idea found by nature itself. That is, when topo makes a covalent bond with a broken DNA, as you may know, then cipro and other topo 2 inhibitors and camptothecin and other topo 1 inhibitors prevent the resealing of the DNA which was temporarily broken. So then you have a prolonged time period where one or both strands of DNA are broken and a big protein is covalently to one end of the gap. Now, the DNA gets all the time inspected if there is trouble, and boy, that is of course big trouble. Repair mechanisms start trying to repair that, but that doesn't work. So then the whole cell goes into apoptosis, and that's of course what you want. So the mode of action of these topo inhibitors is very unusual because you get it. You do not need to block all topos. Only a few of these problem sites is sufficient because you have the cell exploding more or less or emphasizing the damage you made. So every drug should work like that, in my opinion. You just turn something into a toxin, and that's the way how it works. But that's a dream, and we have many others. So the possibilities are enormous, but the difficulties are also enormous. You see here. So as you probably know, designing a drug is not simple. It is not for the faint-hearted. You have to be prepared to spend many decades and get maybe many nice publications, but maybe no patients ever will benefit from your work. But, you know, I come from the Netherlands, and one of the most famous philosophers of the Netherlands is Baruch Spinoza, and he has as one of his beautiful sayings, you know, is do good and feel happy. So that's, you try to do good and you feel happy. It should be slightly modified. Try to do good and feel happy. That's sort of drug design. So it's a slight change of what Spinoza said. Now here you see a family outing in Canada in 1929. I got it from the web. I have not asked the family if it was allowed to show here, so I hope that's forgiven. But the idea is that here's the family waiting for these athletes. But you don't need to be an expert in hurdle racing to see that these are not very well trained athletes. So most likely the family will have to come closer to somewhere here to rescue the broken athletes so they will not make it to the end. And in drug design this can be 10,000 compounds and only one takes all the hurdles and reaches the happy family. So drug design is taking many, many hurdles, and it is very difficult. Nevertheless, we have been working on a large number of targets, as they are called, drug targets, or sometimes possible drug targets. On the left-hand side are bacteria. On the right-hand side are unicellular animals, if you wish, which are they are at least eukaryotes. So here we have Vibrio cholera. This is enterotoxigenic E. coli, cause of many children. It's diarrhea, also traveler's diarrhea. I don't need to say anything about tuberculosis or about leprosy in this building, of course. And then we have worked on cholera toxin and various variants of, and then here the type 2 secretion system I talked about this morning, and the secreted proteases. And tuberculosis, what is red? Red is where I think that other people are working on using our structures, or where we are following up ourselves on our structures. So it's very nice to go to meetings or meet people who say your structure on that and that we really are using for trying to design compounds. That is exactly why I think that structures should be in the public domain, so that the whole wide world can use the naturally occurring structures for design of compounds. So no patenting of structures, in my opinion, for medical purposes. But for the compounds, that is, of course, an entirely different story. So you see we did some work on the malaria parasite, Leishmania, Trypanosoma brucei, the sleeping sickness parasite, and you will see some of them, but of course not all of them, very few I will talk about. And actually the majority, the much of the talk will be on a project which is not on this slide. So topic 1 is cholera toxin, and cholera toxin is an unbelievable molecule. It is secreted by this comma-shaped Vibrio cholera, which you probably know already. The bacterium was discovered by Filippo Pacini in the mid-1800s, published in several Italian papers and forgotten completely. And then Koch rediscovered Vibrio cholera, wrote in German, and the whole wide world at that time was reading German. So, at least the scientific world. So, the cholera toxin was discovered by S&D. If you are complaining about funding, probably S&D was funded by a few $1,000s per year. It was the 1850s in Calcutta, and he did a smart filtration experiment. Make sure that in the filtrate was not a single bacterium. And then he did tests with that filtrate, and they worked just as well in causing the diarrheal phenomena as the bacterial containing solution. So there must be something very small, a factor or a toxin. And then it took a long time before the toxin was identified. At the same time, fully identified and characterized. And then here we have Enterotoxicity E. coli, which secretes two toxins, and one of them, LT, is very similar to CT. All right. So, so the working of cholera toxin is absolutely fascinating, and I show almost nothing about it. But here is AB5. So it is a moonlander-like toxin. The B pentamer recognizes the sugar heads of ganglioside GM1 on the intestinal epithelial cells of human beings. And the toxin of CT and LT do exactly the same. And then there is an enormous complicated process by which the A subunit by itself enters in the cytosol and modifies one arginine of GS alpha, if you like how small modifications can have enormous effects, and that deregulates totally the signaling in the cell. Cyclic AMP gets up way too high, and then ion channels pump out salt, and water follows, diarrhea follows, and in a very serious case of cholera, you might be dead in about 8 hours. That is not common, but it has been documented, to the best of my knowledge. So it is a very serious and complicated disease. What we now are going to talk about is not all the steps inside, many of which are still mysterious, but the binding moment. If this binding event would not take place, and you would take a compound, you would not have cholera. So it's very simple. The beauty of this is it's outside any cell. And in drug, the action, this site of action is in the gut. So the compound has to be small enough to be able to swallow, but that is pretty large, and it has to pass the stomach. But other than that, it does not have to be taken up into the blood. So there is an enormous difference between oral drugs, which have to be taken up to do their work, and non-oral drugs, which can be topical on the skin, inhaled into the lung, or in this case, be given orally. It's still an oral drug, but it doesn't fall under the category of oral drugs because it does not have to cross any membrane. And you even don't want it to cross any membrane. Why would you have the compound end up in the blood? That is not good. If you can't avoid it, avoid it. So, here is the beautiful work of teacher 6, my graduate student in my group in the Netherlands, who around 1990 solved this structure. And you see it's really a moonlander. It is a slightly idealized situation in the gut, so the sun is shining and all that. But so this is intestinal cell surface. In bluish, you see the A subunit. Here in these colors is the B pentamer. And in white are the head groups of the sugars, of the glycolipids. So the sugar head groups of the glycolipids which are attached to the membrane or are embedded in the membrane. So this is the very first contact between the toxin and the epithelial cell. So then many things happen, but we wish to block this interface, this interaction. And we have a very high resolution structure, and we see here a pocket with the pentasaccharide head group of the glycolipid precisely inside. Now, drug design is very often putting a group into a pocket. And in many cases, the way to summarize this, easy to remember, is the best way to design drugs is to put greasy groups in greasy grooves. Hydrophobic groups in hydrophobic grooves, pockets. Here we have a groove, but the natural binding compound is not at all hydrophobic, right? It's a pentasaccharide. And therefore this groove is not very hydrophobic. So now, and it's well known in drug design land, that designing drugs for sugar binding sites is very difficult. And that turned out the case here too. So we made all sorts of variations and we're busy, and actually we didn't get anywhere. But then coffee is very important, right? First of all, for the person who drinks coffee, but also the coffee drinking brings people together. And then they talk. And so this is one of our compounds. We now look, you see this is the P pentamer again, right? The A subunit is on the back, but it's also removed. So here are 5 binding sites. And we were complaining to each other that single small molecules were not doing very well. But then we started drawing a little bit and came up with this molecule. And we looked in the Merck catalog, and you cannot purchase compounds like this. It's strange, but that cannot be done. But why would we be interested in molecules like this? Well, the idea is the multivalency, the pentavalent organization. So we know that they have a pentamer. We know they have 5 binding sites, and they are about 35 angstroms apart, and the non-adjacent. So if we now make this molecule with a finger, this ligand going here, attached with a linker, by a linker to a core, then when this one binds here, the other 4 are already close to the other 4 binding sites, right? So you increase the local concentration of the 4 others dramatically compared to have them free in solution. So this is the pentavalent, and this is then the complex you form. But the idea is that by binding the first one, the 4 others are already nearby, and they don't have to travel that far to get where they should be, that is at the other 4 binding sites. And Erkant Fan, who is the big hero here together with his synthetic group, is that we have the core. He made a linker, and I won't go into any chemistry details. And then here is the finger, the group which goes into the binding pocket. And you see here the only difference is that the linker gets longer. The core is identical, it's very small. The fingers are identical, the blue triangles. But the only difference is the linker length. And you see an immense effect on affinity. So this one is only about 100 times, 200 times more higher affinity than the blue ligand by itself. And do you make the linker longer, it becomes about 1,000 times. Do you triple length linker, it's a 10,000 times. Quadruple linker, it's 100,000 times. That are significant numbers. You know, if somebody earns 100,000 times more than you do, that is a different, in the US that is quite common. Then, not so common, because the number of people high up are, but it's not uncommon, I think. So, it is a significant difference. As a scientist, then you have to ask yourself, why on earth, what is going on? Because the number of people who would have predicted this on this planet before we had done it would probably have been close to zero. So, but at least numerically. And then we thought, if you make it longer, what happens? Now, first of all, the compound became insoluble, which is often the trouble with drug design. So Erkan had to come up with that was an ethylene glycol linker, CCO, CCO, CCO. Here is a guanidinium containing linker and still CCO-ish. And anyway, now it was more soluble. And he also checked the effect of linker length. 2 times it's this high, and then 2 times longer it was, when this number is small, the affinity is higher, is better. So here we have the optimal at 4 linker lengths, and then 6 and H is lower again. So there is an optimum around 4 linker lengths. What is going on? Well, this is the idea that here we have a 1 linker unit, and we look at the distance between these 2 blue groups on average. This is now theoretical, a little bit hand waving, but it is quite, the average we can calculate quite nicely. Oh, sorry. And then you see that on average these 2 blue units are maximally over here, while this is the distance you have to have from this side here to that side there. So here's the tryptophan, there's the tryptophan of the binding site. So this is on average not the optimal distance in the flexible octopus-like molecule approaching the B-pentamer. When you make it 2 units, you get something like this, 3 like this, and you see with 4, the average distance between 2 non-adjacent ligands coupled into this pentamer ligand is just right. When you make it too long, then it's not right anymore, and when you make it even longer, it's even worse on average. So this is actually an entropy effect. So, and it is of course very substantial, because this one is 100,000 times more affinity than a single finger, and this 100 times. So you gain from going from here to here, you gain a factor of 1,000. So this is fun. And we were going along, and we were happy, and all sorts of things happened. And then you have to write a new grant application. And these are these hurdles we were taking, and then all of a sudden there was an enormous hurdle, an NIH evaluation committee. And these were all very bright people, and I won't say too much because this is going to be perhaps on YouTube. So now I have to be much more diplomatic than I usually be at this point in the game. It is a little while ago, but I have my opinion about the soundness of the judgment of that study section at that particular day, at that particular composition. Maybe there was some cholera toxin in their tea or coffee, and they just didn't like the project anymore. Anyway, this was end of project. So you can see yourself whether you would have given us money to continue or not, but this was the end of story. So we switched topic completely. That was a bacterial project, and the drug target was outside the human body. It's inside the stomach, but a mathematician would say it's still outside. Topologically, it is still outside the body. You have not crossed any membrane to get inside the body. Now we go to a protozoan disease caused by trypanosomes. Now many of you know everything about malaria, and you of course have already seen that this must be red blood cells. Now these are trypanosomes, and specifically Trypanosoma brucei here, and it's causing sleeping sickness. And it is an unbelievably sophisticated molecule, organism. It does not go into the red blood cell. It stays in the blood, as you probably know. And we are supposed to have a very sophisticated immune system. And it should take care of this organism in a few days. And our immune system immediately recognizes there is something improper in the blood. And it starts making antibodies by the hundreds or the thousands. And then it starts attacking the trypanosome. And it gets very annoying for the trypanosome. But what is the surface of the trypanosome? The surface of the trypanosome is 10 million, so about 10 to the 7th, identical glycoproteins. So all the antibodies attack this identical glycoprotein. And then after 3 days, the parasite thinks, well, it gets very uncomfortable here. Let's take on another coat. So it changes all 10 to the 7th proteins at the surface for new ones, all identical, but different from the first generation. All the antibodies we make were useless, because the amino acid sequence is different, so that all the antibodies against generation 1 are useless against generation 2. The parasite can do this 1,000 times. Has 1,000 copies with variations of this surface glycoprotein in the genome, and can express one at a time in a particular order. And there are people who have, of course, become very famous of figuring this out. Another amazing property of the parasite is that it can cross the blood-brain barrier. So crossing blood-brain barrier is very, very difficult, even for small molecules. So the parasite is able to do that, and we have no clue how it achieves that objective. So it's a very remarkable parasite in many other ways. And here you see the tsetse fly, which you've heard of, which brings the parasite from patient to patient. And this is a lumbar puncture to diagnose, to see if the parasite have already reached the central nervous system. Because if that's the case, then there is actually only one drug which is useful. And it's also terrible. It causes death in about 5 to 10% of the cases. But if you don't get treated, then you die 100% of the cases. So it is a terrible disease with sometimes 500,000 people per year dying, and then it can go down again, and we are now at a low dip, but you never know when it goes up again. That is the statistics of about a century or so. So there is a need to do something about that. And here we go after very fundamental biochemical process. And that is the synthesis of proteins. And you probably all had this at a certain moment. And we need tRNA synthetases for that. All right, to bring tRNA charged with the proper amino acid to the ribosome. So this is a reaction which is carried out. The enzyme plus amino acid and ATP gets enzyme amino acid coupled to AMP, pyrophosphate disappears, and then there is a reaction with tRNA. In a picture that's easier in a certain way, so the amino acid binds here, and here's the active site, and here we have then, by action of ATP hydrolysis, we have here the an amino acid coupled to the 2' or 3' hydroxyl of ribose at the end of the tRNA. And that then goes, of course, to the ribosome, so that this amino acid gets incorporated when the anticodon of the tRNA binds to the codon of the message. But we only will talk now further about this very first step, or actually this step, where we block pockets in the active site so that we do not get the tRNA even charged. tRNA synthetases, and here we focus on methionine tRNA synthetases, are quite complex proteins. And they have a multi-domain active site region and a very large tRNA binding region. And we'll focus and we zoom in on how to get to that active site blocking. Now, when you do drug design, you have to make sure that you know that your target is really essential. And in the tRNA synthetases, which have been and are quite popular in bacterial drug design, there is a horror story. So there is a story that people targeted a particular tRNA synthetase and worked very hard, purified it, worked very hard for blocking that one. And they got fantastic inhibitors in the test tube. But they made no progress in killing the bacteria. Then the sequence of the bacterial genome became available, and there turned out to be 2 tRNA synthetases for this particular amino acid. So all the work was for nothing. There was no way that with one compound you could block both tRNA synthetases. So you have to make sure, it's called to validate, you have to validate your target. And here it's done by RNAi. And RNAi works very well in T-Bruci. This is a log scale, and this means you knock out the message RNA being made by a very sophisticated procedure, actually. And you see this is the number of parasites growing when you have RNAi not switched on. And here you have switched on, and it is about 4 to 5 orders of magnitude less. So the parasite is very dependent on this particular enzyme. Good. How do you now go from targeting an enzyme to a drug? Well, I'd like to interject one more point. So why is methionine tRNA synthetase a good drug target? Well, first of all, it binds ATP. So it has an adenine binding pocket. And as you may know from work on protein kinases, Gleevec, cancer, and so on, so adenine binding pockets can be very good drug targets. Methionine is quite hydrophobic, so there is a methionine binding pocket. So that could also be a very good drug target, drug binding site. So this tRNA synthetase has several characteristics before you even start doing something which make it likely that you will arrive at a drug eventually. If everything was very hydrophilic of the substrates and therefore of the binding pockets, you have a much more difficult task. Because in this case, the drug has to go through many membranes. It has to reach the parasite in the human body. So it has to go to the blood and then to the parasite. So that is challenging. So we went through one way of MET-RS, is the shorthand for methionine tRNA synthetase, by high throughput screening. And that's a collaboration with the Scripps Research Institute in Florida, where they have a screening center. So there are 360,000 compounds accessible by robot, which then can be delivered to assays in micro format, 1536 well plate. And then one of the key points is to get the good assay. And assays development, as a protein crystallographer, I never realized how incredibly precise that has to be. So in an assay, many of these concentrations are critically important. And here, for instance, is a pyrophosphatase, which removes the product pyrophosphate. And you only need almost nothing of it. But when you have really nothing, then the assay is dead. It's very expensive, so we were very happy that only a teeny tiny bit was sufficient. And each of these parameters which you see here had to be optimized exactly to be good. So this was a multi-factorial optimization. And then we go to the screen, and here you see when there is a kinase glow. So it measures the disappearance of a TP. And here we have a very good inhibitor, and here we have a very lousy inhibitor. And you see that this gives statistics, how good the method is, and every black dot is one of these compounds we are testing. And if they are up here, then they become very interesting. When they down here, they have hardly any effect. So about 1,400 compounds were deemed to be efficient enough. They were tested in a second screen I won't talk about, and eventually also tested on human cells. They should, of course, have no effect. And eventually, they were about 250 compounds were remaining. And now we are looking for compounds which do not, promiscuous means have in previous screens inhibited totally different enzymes, so that's not good. So there was quite a bit of a follow-up. And here is a small subset of the molecules found. And if you look, you can look at this in different ways. You can say, oh, ring. You also can look at it's typically drug-like molecules. You don't see any charge, right? You don't see very large molecules. Also, this one is relatively large. And it shouldn't have chiral centers, because the synthesis has to be not too difficult, certainly not in the beginning, of homologous molecules. They should be easy to make derivatives of, and so on and so on. So this, the art of making this original library has already to take into account what you're going to do with the hits. I understand that there is an NIH only a screening center. I understand that in companies it is so smart that if you do the screen and if you get a hit, a compound which has an effect, you push a button and immediately the follow-up synthesis can start. The robots for doing that synthesis are already ready, so you get small amounts of a variation of compounds. So it's completely, thought through what to do with every hit later, every compound later when it has a hit. We do also crystallography in the lab, and here you see one of the hits sitting in the active site region of our target. And here you see pockets, and we have a few more of these structures, and they still have to be published. So I hope you don't mind, I don't show them all in the greatest possible detail. And, but that is where that project is. So we still have lots to do, and we are determining binding modes. We also want to determine membrane permutation properties. And we'll come back to that in a second. So, another way of getting compounds is starting from the hard work of other people. That's always a good idea. And I think Marseille benefits from the hard work of the Romans and things like that. So benefiting from the hard work of other people is a good idea. So that's also called piggybacking or hitchhiking. And so these bacterial drug design specialists had arrived at this type of molecule, and it's very simple. Ring, linker, ring. You see, it's peanut. And but I've never tested on sleeping sickness parasites. And when you do that, you become. So oh yeah, here about affinities, right? Let me see. Just test how much you know about drug design. It's a very simple question. Don't worry about how good you're doing. Who knows about the Lipinski rules? Okay, fair enough. So that's so none of you is involved in a drug design project, right? So okay. So what is a good affinity of a drug for a target? That's very simple. You go to a drug design meeting and you see people walking like this. Those have millimolar compounds. Millimolar, that's not good. Then you have people who walk somewhat like this, they have micromolar. Then you have people who walk like this, they have nanomolar compounds. Those are the good ones. And then some of them, they don't walk. They are higher up, they fly. They have picomolar compounds. So it's very easy at the conference to know who has the good compounds. So here we walk like this, it's very good. Single digit nanomolar is it called. So that is a very good inhibitor. But we haven't done very much, except Fred Buckner Fred Buckner has set up in his group with Renee and Rob has set up beautiful assays, and this was one of them. So you can understand it was very stimulating. But then do these compounds work as we think they work? And that is called chemical validation. So if the compound works as we think it works, there should be a correlation between the activity on the enzyme in solution and the activity on the parasite. And that is sometimes very disappointing, because a compound may work for totally different reasons than you think. Here we have every symbol in black is a different compound, the blue one. And this is the thermal shift. So that is the degree by which the compound stabilizes the thermal stability of a protein. And the more tighter a compound binds, the more stable the protein becomes, and the melting temperature goes up. So that is, here's about a 12-degree shift in increase in melting temperature. And this is a very low shift. And you see, this is the effect on the parasite. So there is a tremendous correlation with a R square of 0.91, and that was sufficient for a good study section in NIH to give us money, you see? So that is very solid data. What is this? This is for Trypanosoma cruzi. So this is Trypanosoma brucei here, the sleeping sickness parasite in Latin America. A very important disease is caused by T. cruzi, Chagas disease, and it's a totally different, it's a related parasite, but the disease is totally different. Here you have more compounds. There is no drug, but you see we have here quite a few over time, which now we start modifying the compounds and start getting more information. We also would like to know, how do they bind? We could never co-crystallize the compound. And so we started with a crystal with methionine bound. Now, who of you are crystallographers? 1, 2. All right, 3. Okay, good. But not many, right? So many not. Yeah. So all right. So this is okay. Here we have. You see, this is methionine bound in the active site. If you block that binding site, you don't have enzyme activity. What we first showed, just that the crystals are very interesting. If you add ATP, you get a reaction in the crystal. So the enzyme is active in the crystal. So that's nice. It's not, we have a really fully active protein. And now, this is made by a postdoc, Cholera Toxin from Singapore, and he likes jokes. So, and I like his jokes, so here's his joke. So this is, I think, him. And these are crystals after you add compounds in the imidazole to the methionine containing crystals. And immediately the crystals start getting shattered into pieces. But he's from Singapore and he never gives up. So he sits there. If you see him sitting, then you know he's busy. And he takes the crystals, I don't think with a fishing rod, but with something else, and freezes them at -80 degrees immediately. And the compound needs only 30 seconds or less to fill all active sites, in most cases. Here he looked at subunit A, and nothing had changed. The methionine was still there. Experiment completely failed. But he is a smart cookie, as they say in English. So he also looked at the other active site, and there was the compound. And I made the best mistake in my life ever. When he did this the first time, I said, Cheerio, it was fantastic, but I don't think you will ever be so lucky again. And I still have some holes here in my body. The eyes by which he looked at me were just penetrating. And this time he didn't laugh, and I had made a fatal mistake. But the good thing was, he was more inspired than ever to do it again. And he has done it maybe 1,000 times ever since. So I have apologized 1,000 times, and we are now good friends again. So, so this has been very successful. And what happens is that one subunit is packed, and it cannot change conformation. The other subunit is sort of making interactions, but the active site is wide open. And there all sorts of things shift when the compound binds. And here you see one change, and I apologize, it is, I couldn't find high-resolution versions of these pictures, but here you see the methionine in a small pocket, fits beautifully, and here's one ring. Remember the compound? Ring, linker, ring. So one ring sits here where the methionine binds, but it has a much larger pocket. But here you see the linker, right? And here's the other part. And now we take another view. Here's another Here's the other one linker. This has another pocket, which was totally absent when methionine was bound. So the protein adapts to the compound present, so it provides two binding pockets. One of them we expected, the methionine, the other one was totally unexpected. So then we do an in vitro toxicity, no toxicity. Mouse acute toxicity, mouse keeps happy. And then we try to do some effects on live mice as oral drugs, but the time is moving forward like so often. So I won't go into details here, but it had some effect when you gave it with a pump implemented in the mouse, and you can see that it takes away the parasite living in the mouse. But the problem is that if you give it orally, not high enough concentration in the blood. And that is this blood-brain barrier, and there are assays for that. So it's very simple in a drawing. It's much more tricky to do it, but René has it fully under control. So here we have a cell layer. Here we have one compartment. Here we have the other compartment. You bring the compound here and you measure over time the appearance of the compound in the other compartment. And this has even a drug efflux pump expressing as well. So it's a pretty good imitation of the blood-brain barrier. And then if the bar is high, you go through the membrane very well. And here is our super high affinity compound, and it is a total disaster. So this compound has high affinity, but doesn't cross membranes. So that's where this one is. We have a fantastic set of compounds as inhibitors, beautiful, but we have to improve membrane permeability. And so we came up with an idea called 1356, and you see it's going through the membrane. This is one which goes through, this is a test compound, nothing to do with the project. It goes very well through the membrane, and this is almost going as well. So what is the difference? The difference is that this was this 1312, and we have only made a minor change here, you see. And if you have had chemistry, you see that is a urea compound. And if you talk to people in pharmaceutical committees and you say, we changed a compound so that it has now a urea compound in order to get better membrane permeability, they look a little bit sad at you when you say that is exactly what you should not do. But then I look a little sad at them because this compound is much better permeable than that one. So it goes against the folklore, right? So we understand a lot in science, right? We have now the Higgs boson, and we have quantum mechanics, and we understand everything. No, we don't understand how well a small molecule goes through a membrane. It has to do with water, and everything that has to do with water is not very quantitatively understood in general. So the membrane penetration properties of a compound are actually a fantastic challenge for theoretical chemists and fundamental biophysicists, etc. But these compounds now has a much better, excellent membrane passage, but we lost a factor of 50 in affinity. You see, and that is the challenge in drug design. You optimize one property and then you lose in optimizing with respect to another property. So, I have to stop here, but we have now, from this change, we have IC50s of 100 to 200 nanomolar, which is not all that good, and also the EC50s, and they have reasonable membrane penetration properties. So, actually those people walking, I was at Nijmegen on Tuesday, and they had picomolar inhibitors. So for a while they were, in the air, high up, and then they started doing membrane permeability, and they are now all the way back to Earth because there was no membrane permeability enough. So it's a hard job, this drug design, and so that's how it is. So here in our case, it's slightly exaggerated. We hit first the permeability barrier, but then we brought this down. But let's say the enzyme assays, that then went up. So it's it is even much more complex than the hurdle race. Every hurdle is actually different. All right, so what is the point? I strongly encourage you to be involved in drug design projects if you can, because drugs are often absolutely wonderful compounds. If your child has a very bad ear infections, fever and everything, and you give one injection with good old penicillin, at least this was 1970, in 2 hours time the child is happy playing again and all is over. So these are really wonder compounds. You all know, of course, about drug resistance problems, so this story about actually true in 1973 in Nairobi, I found it unbelievable. Whether that is still so easy today, I'm not a medical doctor. But to get to a drug, it takes a fine balance. Is, for instance, very simple. You have to cross membrane, so you like to be a little bit hydrophobic, but you also have to be sufficiently water soluble. Otherwise, the blood doesn't transport it to the site of action. You should not be converted into another substance by human enzymes, and we have plenty of those. Clearance from the blood. It should not cause bad effects on the unborn child. It should not have effects on the DNA. Of course, the toxicity should be very limited. And if you talk about tropical diseases, it has preferably to be stable at high temperatures, and it should have an ultra-low price. So drug design for low-income countries is even more difficult than it already is for high-income countries. But that's the story I have to tell you, except for who are all the heroes. Well, this is this multivalent inhibitors on cholera toxin. And here you see Erkang Fang and Zhongsheng Zhang as the major people. And many others have been helping in several ways. And here the tRNA synthetases. Here are a variety of heroes. Christopher Linde in target selection, but also in chem informatics and chemical modeling. Protein production by a whole group headed by Wes van Voorhis. Chemical synthesis, the group of Erkang Fan, the same group who also was critical in the toxin story. Assays, a fantastic group of Fred Buckner, Rob and Renee. Beautiful screening results in Florida. And here are the crystallographers, and I like really to highlight Shoyo Ko, who did his marvelous work with soaking compounds in pre-grown crystals. And financial support from various sources, and I thank you for your attention. Questions?
I have two questions. The first one is that, have you gone through all these barriers and everything as a drug design individual? How do you know when to stop, maybe on a particular project that, oh, I think this, how do you know when to stop? That is one. And secondly...
Wait, maybe we try to stop that, answer that first. I'm always very good in listening, but then I forget what your first question was. So, so when do you know where, how to stop? Well, there are certain criteria. And in malaria, it is, there is a fantastic organization called MMV in Geneva, Medicines for Malaria Ventures. So you can go to the MMV website and you see that they have a number of projects, and you see a whole set of criteria going to the right. That is the progress these compounds have made in the drug design pipeline. So that means that is really the hurdle idea. So they have taken one more hurdle and one more hurdle and one more hurdle. And when they have a low toxicity, when they have no effect on the human heart test, not real human heart, but a homologous test, so are not converted by a whole set of human enzymes, when they have high enough blood levels in rats, and so on, then at a certain moment the MMV says, now we're going to test on healthy human volunteers. There's not so many yet. Then they go on into clinical trials, and hundreds of millions of dollars have to be have to be made available. So there is a well-known, for each disease that is different to a certain degree, and so there is a well-known set of conditions, which I don't know by heart, where the compound has to fulfill in order to go into these final stages, where you have to show eventually in selected patients that you cure patients, right? So I cannot answer your question simple, but if you go to the NMV website, you may get a very good idea of how this is done for malaria in practice. Do you still remember your second question?
My second question is, it's not really personal, but I just want your personal opinion concerning the emergence of resistance in infectious pathogens, and having gone all through all these painstaking ways of drug design, then what do you feel?
What do I feel? Well, it's something which is very close to my heart, and I really thank you for asking this question. So we have seen that my last 40 years, more or less, have been devoted to trying to stimulate the design of drugs against tropical diseases, and I've tried to do my little bit myself. And I'm very happy, actually, I forgot to say, that we have some very promising compounds for Chagas disease. And we are now curing, we is Fred Buckner's, but it's our collaboration, are curing mice infected by the Chagas parasite, the trypanosome. And my happiness is actually now dependent on how happy these mice are jumping around. It's very amazing that your well-being is now depending on the happiness of mice. But what is happening now with them, I alluded to that already a little bit when I talked about my daughter and penicillin in the 1970s. There is worldwide, and the WHO, I think a month ago, made an enormous announcement that this is a major problem. If you read Nature, and I strongly recommend that you take a lifetime subscription on Nature forever, and read not only the difficult parts, but also the early parts. And the same, actually, if you can afford it, you should do for science. It gives you a broad outlook on all aspects of science. And the nice thing, compared with all the horror stories in the regular journals, I mean the regular newspapers and so on, is that much of the news in science and nature is very positive. There are new developments, new drugs, new discoveries. It is positive reading, for me, counterbalancing all the negative news, which is, for some reason, always in the highlights. If you took Nature of a month ago, you would see a special section of about 7 to 8 publications totally devoted to your question. What are the problems with multi-drug resistance? What are the most important, it's all bacterial resistance. What are the most important bacteria where resistance is occurring? Which are the companies working on it, and it's getting a smaller number of companies. What are the methods we could be using, etc., etc. Of course, it's not a completely comprehensive coverage of everything, because that would be books. But still, it's a very good way of getting into that. The problem is, of course, that if you have broad spectrum drugs, you will earn more money, because you can use the compound for treating many people. Many people buy your compound, so you become rich, and therefore, as a company, you put in first money and you get the money back. That's, of course, the capitalist system. Other systems, non-capitalist systems, have not produced many drugs. I'm not a great capitalist myself, but a fan of capitalist system, because it has very cruel features. But the competition does get things done. If you have now highly, like the cholera toxin here, right? That would be a compound. Imagine that it would be cheap, You have a compound which fulfills everything. It only will be useful for treating cholera patients or preventing cholera. That is not what they call a blockbuster, right? So how do you get that made? And that is, of course, a major problem. And some pharmaceutical companies, when it's not too expensive, help out. River blindness is a beautiful example where Merck is doing a fantastic job in making ivermectin, an N at the end, free available for, or very cheap available for, no, I think it's even free for treatment of river blindness, and that is fantastically successful. So don't get me wrong, there are beautiful things done by pharmaceutical companies as well. But the rules of the game are thus that if you put in $1, you have to get more than $1 out at the end, because otherwise the company has to stop. Whether you own it or I own it, there is no way around it. So the number game is very important. So how do you now get drugs for cheap drugs, when you have small patient numbers? And that is a problem where maybe the capitalist system will not work. We have now drugs for certain cancers where only very few people benefit from benefit from the drugs. The prices are in the tens of thousands of dollars per year. Yeah. For cholera, if I would have a beautiful drug with $10,000 per year, it will not be used very much, right? So there is, your question raises very many non-scientific questions. Because it's scientifically so difficult, it costs money, and lots of money, so maybe the world should be thinking. I've heard certain people who are deeply involved in American scientists, deeply involved in drug design, doing fantastic things. They say maybe for this bacterial resistance problem, we need to have sort of a worldwide Manhattan Project. You know, the Manhattan Project for the atomic bomb, which let's now do it for something which is an antibacterial bomb or something like that. Maybe we should do that, which was, of course, not a business model, right? It was funded by the population. So I'm just throwing out a few things. You just pushed a button in me where I get going maybe a little too long, but I apologize for that. Christian?
It's surprising that these compounds don't bind to human material synthetic. They're so different.
Wait. I didn't say that. I didn't say that at all. I said multiple times that maybe they do not affect human cell lines. They hit mitochondrial methionine tRNA synthase from human beautifully at the 10 nanomolar range. So that is a drawback of these compounds, but wherever we have tested them, the mice is jumping around. We think that the reason is that it's the mitochondrial tRNA synthase. So the compound has to, from the gut to the blood, from the blood to the human cell, but then in the human cell, from the cytoplasm to the mitochondria. Maybe, I don't know for sure, maybe the last step is not taken. And anyway, in the 5 cell lines, I think, which so far have been tested, and the many mice, not many, but the mice which have been tested, the compounds, this class of compounds, has no obvious toxic effect. That's my answer. But you are totally right that you were amazed, and you are absolutely right, there is no selectivity at the enzyme level.
Is it effective against other parasites? In this case,
if
it's not so selective, then what about malaria and so on?
No, yeah, we have tested that. That doesn't work. But the T. brucei and T. cruzi, that seems to work. But T. brucei, the sleeping sickness parasite, you have to reach the brain, which is a whole different game than reaching macrophages. You see, so that is so there is a dual potential use right now. We believe that it is more. We have a bigger chance in Chagas than in T. brucei.
And did you go back to bacteria?
No,
when they look for bacteria, they look for corals.
Yeah, but
what about the other way?
But the bacteria have been studied so thoroughly that that companies have been formed and have gone and studied it all.
I mean, in your lab, did you put with E. coli staph just to see?
I'm certain that certain compounds didn't work at all. But,
is the use of chemistry
now you go into a field I know very little about, except that I know that you bring compounds together and click and you have a new one. Yeah, so that's roughly my full understanding of click chemistry. We have not used that yet, but we do discuss it. So we have every monthly group meetings and the chemists. So the crystallographer comes, the Shoyo says, I have here a pocket, there's another pocket, you should use click chemistry. And then the chemists look a little sort of, he's a little simple-minded, but he's pretty bright, I would say. Very bright. But you have to have the right, it sounds as if you know more about it, much more about it than I do.
Okay, c'est une question. Thanks again for the talk.